All right, my G Block class, I'm going to be talking to you today about amniocentesis and karyotypes. And this is kind of an amazing technology that probably many of your parents actually went through, and you just didn't realize it. And the whole point of doing an amniocentesis is so that you can create a karyotype, and then a qualified technician can look at the karyotype and tell whether or not there are problems. What kind of problems are you talking about? Problems with a developing fetus. So an amniocentesis is something that's done on a pregnant woman. So let's take a look at what's going on with that. I'm just going to import a picture in here. Let's see, where is it? It's right here. Looks a little creepy. So let me explain to you actually what's going on here. You obviously can see that we have the developing fetus going on in here, and we also have this thing that allows you to carefully see inside, see what you're doing if you're the doctor who's performing this technique. And you can see, frighteningly enough, that there's this gigantic hypodermic needle that not only has to penetrate the abdominal wall of the woman, but then has to penetrate through her uterus and then into the amniotic sac. The amniotic sac is what is surrounding the developing fetus. Now, what's interesting that many of you probably remember from my screencast about Dolly is that a fetus, when it's developing, will shed a whole bunch of its cells. They're sort of just going to fall off, much in the same way that you shed skin cells, except that these cells, when they're shed, very much stay alive because they're hanging out in an amniotic fluid which contains all the nutrients and all the perfect conditions in order to allow something to be maintained. It won't necessarily grow, but it'll certainly not disappear. So you would be able to go into that amniotic fluid and you would find a lot of live cells that are genetically exactly what the cells of the fetus are. Okay? They share the same DNA because obviously they'd be body cells from that fetus. So you extract it, and usually you take out about 20 mils, which is about 4 tablespoons or so of this amniotic fluid. Now don't worry, that in about 2 days that amniotic fluid is completely replaced, and in fact in about the same amount of time the puncture to the amniotic sac and the puncture through the abdominal wall and through the uterus are also sealed up. So as long as the doctor is careful not to actually poke the needle into the fetus itself, it's a very safe procedure. Of course, you got to have that ultrasound there so you can see what you're doing. You wouldn't want to do this blindly. All right, so you extract this 20 milliliters of amniotic fluid, and let me just go to the next page here. So imagine you have here a test tube, and it contains the amniotic fluid, and it also contains a whole bunch of the cells of that particular fetus. What you want to do then is separate it out so that all you have is the cells. And in fact, you put it into this funky, it's not so much a bottle, I mean it does look like a bottle, but it's a tissue culture flask is what they're called. And what they do, these tissue culture flasks, you can actually grow cells on the bottom. And you grow them to what's called confluence, which means until they're touching. So you would take out those fetal cells, you'd put in the right conditions into this tissue culture flask, and you would grow up a whole bunch of these fetal cells here. You'd get rid of the rest of the fluid, you don't really need it, so this whole thing disappears once you get it into this tissue culture bottle. And you grow them up, and then just before you're going to extract them, you actually pour in, because this is an opening right here, a chemical called colchicine. Colchicine is a plant extract. I can't remember. Bonus points to whoever tells me what plant colchicine is extracted from. It's like, yeah, saffron. Oh, sorry, no points for anybody. It's from a saffron plant. And what colchicine does is it's a chemical fixative. You might ask, what the heck does that mean? Well, in this case, what the fixative does is it allows the cells to grow and go through interphase and then go through the S part of interphase. So you have the duplication of chromosomes that would occur. Actually, would you necessarily have that? Yeah, I guess you would have the duplication of the chromosomes going on. And then what you would have is just before it's going to undergo mitosis, or actually it even gets to the phase where it goes through, no, what am I smoking? Yeah, I'm sorry. So it goes through prophase of mitosis, so all the chromosomes get condensed so you can see them very easily. And then you throw in a special stain so that you can more easily see those chromosomes. And then the fixative locks it up at that point. So instead of going through metaphase, anaphase, and telophase, it stops at prophase, or it stops just before metaphase. So that means all the chromosomes are condensed and visible to somebody looking through a microscope. And at that point, you take a photo. And what's kind of amazing about this is that you literally just take a photograph under a microscope of the chromosomal spread, and it may look like something like this. A whole bunch of chromosomes hanging out inside of a cell. There'd be no nucleus, right? So, because it would already have broken down as a result of being in prophase, and it'd be hanging out just like that. Okay? Take a photo of it, and then you print out that photo, and you literally take a pair of scissors and start cutting out. You blow it up, obviously, so you can see it more clearly, but you then cut out each of the chromosomes. And this is what it looks like when you're done. There you go. These are the chromosomes. They've been stained. That's why you can see the sort of light and dark banding associated with them. You arrange them in order from the longest ones to the shortest ones. You pair them up so they're with their homologs. And of course, you do have to do some techniques so that you don't, you're just looking at one of the chromatids. You don't have to have both sister chromatids at this point. But you have one from mom, one from dad. And you can see here that you should always have pairs, right? So every single one of these from chromosome 1 through 22 is paired up. And in this case, however, the sex chromosomes, right here and right here, they are not paired up because in this case it is a boy, a little baby boy is being created here in its mommy's womb. So everything is in order. This would be a great thing. If you got this back, then that means you have a clean bill of health and that your child is not going to have any sort of chromosomal abnormalities which could lead to some serious syndromes. So here's a male. Let's take a look at a female. Let's see. Looks like this is my female. There we go. Here's a girl. You can tell she's a girl because notice here, there's no Y chromosome. There are just two X chromosomes going on here. So pretty cool, huh? This is, again, a healthy baby girl that we got going on here. And hopefully that's what you guys will all have. But let's see what happens if we have aneuploidy, like we showed in yesterday's, like I showed you guys in yesterday's screencast, where instead of having the proper number of chromosomes, you have an improper number of chromosomes. And that looks something like this. Oh boy, where is it? Boom. Okay. Now I want you to go through and take a look at this. Because if you look carefully, this is fine, this is fine, this is fine, this is fine. These are all paired up and everything. And then boom, we get here to chromosome number 21. And there's 3 of them. So clearly this was an issue where the egg, and this is always on the egg side of things, it's not on the sperm side of things, the egg had a non-disjunction occur so that 2 chromosome number 21s from mom entered into the fertilization event, so the egg had 2 chromosome number 21s, the sperm had 1 chromosome number 21, and you have a child then with Down syndrome. I'm sure we all know people, or at least we've seen, if we haven't met people who have had Down syndrome. We all know that people who have Down syndrome, they can be perfectly happy folks who can be productive members of society. So in fact, Down syndrome, while you may think of it as being severe, is probably the least severe of all the syndromes that one could get. That's in many cases, you would make decisions when you get the karyotype back. There are some parents who might decide that if you have a child with Down syndrome, they'd want to terminate the pregnancy, i.e. get an abortion. But others would say, you know what, having a child with Down syndrome, I'm perfectly fine bringing him or her into the world, and because I know that she can be a productive member of society, so why not? Obviously, that's a decision for you to make, and something that you will have to make sure you make that decision if you decide to be a parent someday. So Down syndrome, like I said, is probably the least severe. Let's take a look at something that maybe is not so great, and this would be a child who has what's called Edwards syndrome. And if you take a look here, let me just point out, everything's okay. This happens to be a boy, by the way. You can see X and Y. Except when we look right here, chromosome number 18, we have a trisomy, and in fact, that's a good word. Trisomy, 3, right? Somy, chromosomes, number 18. So it's called trisomy 18. In the same way, by the way, that Down syndrome is sometimes known as trisomy 21. Okay? Having 3 again is an important thing there. So trisomy 18, also known as Edwards syndrome. And let's take a look at a child with Edwards syndrome. You'll see it's a little bit frightening. And this is what I was warning you guys about earlier. Here is a kid with Edwards syndrome. Brutal, right? You can see there's serious formation issues, malformations. The legs are all twisted there. Usually there's severe mental retardation. Oftentimes the child won't live for more than 3 months. And there are actually even more severe pictures. If you really want to look, and I didn't want to, this is actually a relatively tame picture of Edwards, but you can certainly go online and look up Edwards syndrome and see some of the really just pathetically sad situations where children are born with this disease, with this syndrome. Okay, let's quickly get away from that one. So we also have another syndrome called Patau's syndrome, or Patau. I'm not quite sure how it's pronounced, but in this case, you can see here we have the eggs. The egg is screwed up again, unfortunately, and has led to a non-disjunction where you have a trisomy 13 going on over here. And like I said, it's called Patau's syndrome. And Patau's syndrome, I would even say, is potentially more severe than Edwards. Now, the picture I show you isn't going to look as severe, and that's mainly because I'm only showing you the shot of the child's face or head. But you could imagine if your face looks like this, the rest of your body probably isn't going to do so well either, and certainly your skull and your brain is going to have some serious problems here. Again, most people who have a Patau syndrome or Edwards syndrome, typically the pregnancy will terminate itself. You'll have a miscarriage before it even gets this far. In some cases, for those unlucky few who actually go through full-term pregnancy and the child is born, the child will only live for a few years. There's some cases where a child who has Patau syndrome can live for a longer period of time into her teens, but for the most part, it's a death sentence for these children. Now, the question you might have, and it would be a good one, is why am I blaming the egg? And the reason why these things always happen to the egg cells is that a woman is born with all of her eggs in suspended animation. They haven't fully undergone the meiosis events to develop into an egg cell. However, they've gotten a good way towards that direction. And the problem you have here is that if you have your eggs sitting around in this suspended animation for a long period of time, say 25, maybe 35 years before the egg is actually released into a fallopian tube and then fertilized by a sperm, that's some serious aging. I want you to think about anything that you would own for 25 years. It's going to wear down, it's going to break down, it's not going to do so well as compared to its first few years. And so that's why amniocenteses are often done for people who are 35 or older, because at that point your egg cells are having a hard time completing the division stages of meiosis, going through meiosis 2. And you can imagine if you're having a hard time going through those last few stages, a lot of mistakes can be made. The spindle fibers, which are the main reason why aneuploidies develop or non-disjunctions develop, they are not going to do as good a job at attaching to the kinetochores of homologs or of the sister chromatids and pull them apart properly. So when you hit this age, 35 plus, it's really highly suggested that you do the amniocentesis to be sure that you can at least make a decision or be prepared for what you might do with your pregnancy if your child, if it turns out that your child has one of these syndromes. All right, the last thing I want to pass on to you guys is that you actually can have non-disjunctions. Oh, sorry, to go back through here. You can have non-disjunctions associated with the sex chromosomes, and this is kind of interesting. So can you imagine a situation where you could have 3 X's, or maybe just 1 X chromosome, or you could have 1 X and 2 Ys, or 2 X's and a Y? That really makes a big mess of things. Now, and you know how I said earlier that trisomy 21 or Down syndrome was the sort of the most mild of the syndromes? Well, in fact, these sex chromosome abnormalities are actually probably truly the least debilitating of them. In fact, it's very likely that there are peers of yours at this school who are XXYs or just single X folks, because in many cases you wouldn't even notice. In extreme cases you would notice some things, but for the most part, if they're walking around our campus going to school at Choate Rosemary Hall, it's sort of, they don't even know about it until, these people don't know about it until they try to have children themselves, and then they have a hard time having children because of the extra or too few sex chromosomes. All right, that's enough. See ya.